Hello guys, now let's begin with the first chapter in the pathology, that is cell injury. Okay, now in this video I will be mainly concentrating on what are the causes of cell injury and the types of cell injury. Okay, there is something called as the reversible cell injury and irreversible cell injury, and what are the changes that are going to be seen in the cells which are undergoing reversible and irreversible cell injury. Okay, now without any further late, let's begin with the topic, or I should say the chapter, cell injury. Now, my first question is, what is the most common cause of cell injury? Okay, what is the most common cause? Why a cell is getting injured? The most common cause is hypoxia. Now, my question to you, what exactly is hypoxia? Hypoxia means what? Hypoxia means decrease oxygen to the tissues. Okay, now there is less availability of the oxygen for the tissues, whatever might be the reason, because of some blood vessel blockage or because of some reason, there is hypoxia, and this hypoxia is the one which is going to cause damage to the tissues or the cells. Okay, the most common cause of hypoxia is next question, most common cause of hypoxia is ischemia. So, what exactly is ischemia? Ischemia means decrease blood flow. Okay, decrease blood flow to the cells or decrease blood supply to the tissues is called as the ischemia. Now, why ischemia? Might be because of there is some blockage in the blood vessel. Okay, so hypoxia is because of ischemia. Ischemia is not because of hypoxia. Ischemia will lead to hypoxia. Decrease in the blood flow lead to decrease in the deliverance of the oxygen. Ischemia. So there are other causes of hypoxia also. Okay, but before going to discuss about the hypoxia, I want you to know which cells in our body are very much sensitive to hypoxia. There are certain cells in the body, human body, which are highly susceptible to hypoxia. If there is hypoxia, they will die. So next question, cells most susceptible to hypoxia. Okay, cells most susceptible to hypoxia. Answer is neurons. And cells most resistant to hypoxia is fibroblasts. So which means fibroblasts, whatever you do to them, you just expose them to the hypoxia, still they will be living for a longer duration. Which means they are more resistant to hypoxia. And which cells will undergo first degeneration, first death? It's the neurons. Usually neurons will die within less than 5 minutes. Okay, if you expose them to hypoxia, within the 4-5 minutes they will die. Now let's discuss about hypoxia. Okay, how many types of hypoxia are there? Actually, for exam purpose, I want you to know 4 types of hypoxia. Okay, 4 types of hypoxia. What exactly are they? First type is called as hypoxic hypoxia. Okay, hypoxia, which means there is a hypoxia outside. Okay, there is hypoxia outside, which is leading to hypoxia in the body. For example, whenever you go to high altitudes. Okay, whenever you go to high altitudes. So there is hypoxic environment outside, so that you cannot get the proper amount of oxygen. Okay, so hypoxia is seen with high altitudes. The second type is anemic hypoxia. What exactly is anemic hypoxia? Outside is okay, there is no problem with the tissues, but the problem is with the blood. See, in the blood, not enough carrier molecules are there, not enough hemoglobin is there. So hemoglobin is not properly present, means who will deliver the oxygen to the tissues? Who will deliver the oxygen to the cells? It's the hemoglobin. Whenever the hemoglobin is not sufficient enough, that can also lead to hypoxia. Okay, outside everything is normal, even the tissues are having no problem, but still they are getting hypoxic because of some blood related reason, that's anemia. Okay, so anemic hypoxia, there is a decreasing carrier. Okay, there is decreased carrying capacity. Okay, anemic hypoxia. See, anemic hypoxia, it is seen in which conditions? First of all, there is anemia that can lead to anemic hypoxia. The second reason is it's also seen with carbon monoxide poisoning. Okay, carbon monoxide poisoning. See, carbon monoxide, we know carbon monoxide have better affinity with the hemoglobin. It have almost 210 to 240 times more affinity for the hemoglobin. So, imagine there is this hemoglobin molecule. If you present this hemoglobin molecule with oxygen and carbon monoxide, the hemoglobin more binds with the carbon monoxide. So, there is no place for the oxygen to go and bind with the hemoglobin. So, in the areas where there are burns, okay, for example, there is a fire accident where there is lots and lots of carbon monoxide, carbon dioxide is there, carbon monoxide is also there. So, the person, if he inhales more and more carbon monoxide, so carbon monoxide is going to get bind with the hemoglobin. So, there is no space for the oxygen to go and bind with the hemoglobin. So, that can lead to anemic hypoxia. Okay, so problem is where? Problem is in the blood. Now, the third hypoxia is stagnant hypoxia. So, stagnant hypoxia, it is seen in which conditions? See, there is no problem outside, everything is okay with the environment, normal environment hai, and there is no problem with the blood also, hemoglobin is properly there and everything is normal, but the pumping action, who is the pump? It's a motor pump, is our heart. There is problem with the heart, so it's not contracting properly, might be because of congestive heart failure, because of any other reason. So, there is more stasis, there is a stasis of the blood is getting stagnant. So, no proper oxygen delivery to the tissues because of the motor problem, the heart pumping action is not good. So, stagnant hypoxia, it is seen in conditions of heart failure. Okay, so no proper perfusion to the tissues and no proper drainage from the tissues, so leading to the stasis, and that can lead to hypoxia. And the fourth type of hypoxia is histotoxic. Histotoxic hypoxia. Now, in the name itself it's there, clear. Histo means tissues, toxic means there is some problem with the tissues, there is some problem at the level of tissues which is leading to the hypoxia. See, this histotoxic hypoxia, it is seen with cyanide poisoning. Cyanide poisoning. See, you know this chemical, right? The cyanide, what it will do, whenever the person is taking the cyanide, it is going to inhibit, the cyanide inhibits electron transport chain, complex 4. Okay, so in the inner mitochondrial membrane, there is something called as complex 4 of the electron transport chain. This complex 4 is inhibited. When this complex 4 is inhibited, the oxygen is not going to be used for the production of ATP. Okay, so there is some problem at the level of tissues. The tissues cannot use the oxygen. Outside oxygen normal, the heart is normal, the hemoglobin is normal, everything is normal, but the problem is at the level of tissues because the tissues cannot use the glucose, the tissues cannot use the oxygen for the production of ATP, for the oxidative phosphorylation. So, this is histotoxic hypoxia, seen with which poisoning? Histotoxic hypoxia, it is seen with cyanide poisoning, where the complex 4 of electron transport chain is inhibited. So, with this, we have discussed about the types of hypoxia. Now, after this, let's discuss about the cell injury. Now, let's enter into the main topic of the cell injury. See, whenever there is stress, okay, I am giving stress. Okay, so there is this one cell, look, there is this cell with the nucleus. Now, I am just giving it the stress, I am keeping it in the stressful environment. The stress may be of different types, maybe microbiological stress or chemical stress, immunological stress, okay, or it can be physical stress or anything. Now, I am stressing the cell so that the cell is in not normal environment. Now, it's in a stressful environment. Maybe there is a radiation exposure, chemical exposure, microbiological stress, immunological stress, some toxin mediated stress. So, at the end of the day, the cells are being totally attacked, or you can understand something like that. Okay, there is stressful environment. Now, whenever there is stress, yes, of course, cell will undergo injury. Okay, the cell is getting injured. Now, whenever the cell is getting injury at the stressful environment, first what the cell will try to do? The cell will try to undergo adaptation. Okay, whenever there is a stress or injury to the cell will try to undergo adaptation, that is called as cellular adaptation. Okay, cellular adaptation. Now, even you guys, for example, if I am giving so much work, okay, if I am giving so much work, what you will try to do? You will try to, okay, sir is giving so much work, so you will try to do it. But I am giving the work beyond your capacity. So what you will try to do is you will try to adapt. Okay, so that you will stop watching all the movies, you will stop like going for the sports. So continuously you have adapted yourself to do the work, to manage that stress. So in the same way, when I am giving the stress to the cells, even cells will try to adapt. That is called as cellular adaptation. We will discuss about all the cellular adaptations in a different video. But the stress is too much. Okay, overwhelming stress is coming. Now cells will say, I'm done, I can't take this much stress. So cells will first undergo reversible injury. So after cellular adaptations, what happens is, if there is stress, even the stress is continuing, then there will be reversible cell injury. There will be reversible cell injury. Now in reversible cell injury, see, certain events will happen, but in the name itself it's there, that injury is going to be reversible. Okay, if you just take out the stress, if you just remove the stress, again the cell will go back to its original function, shape and normal. It will become everything absolutely normal. First, there will be reversible cell injury, but still stress is continuing. Now in this condition, the cells will say, I can't take this much stress and I am done. So that reversible cell injury will continue as irreversible cell injury. Okay, irreversible cell injury. So now in this class, we have to see what are the differences between cell injury and irreversible cell injury. See, in reversible cell injury, I have explained you. The cell will go back to its normal function if you remove the stress. But in irreversible cell injury, in the name itself it is there, irreversible. The cell injury is irreversible, beyond repair. So cell will 100% will die. So whenever there is irreversible cell injury, cell will undergo death. That is cell death. Okay, cell death. So there are different types of cells in our body. Different types of cells will undergo different types of death. Okay, for example, just for understanding purpose, I am telling you, there are different types of people in this world. If they want to die, they will commit suicide in different ways. Someone can stab themselves, someone take poison, someone can hang. So there are different ways of death. In the same way, there are multiple different types of tissues in the body. They will undergo different patterns of death, different process of death. Okay, which are different on histology, which are different in the molecular, the pathophysiology of their death is different. So cell death will occur. What are the different types? One type of cell death is called as necrosis. Second type is called as apoptosis. And there are even other types, subtypes like necroptosis and pyroptosis. Okay, necroptosis, pyroptosis, all these are the different types of cell death. Now when you will have the cell death, whenever there is irreversible cell injury. Okay. Now first let's begin our topic with reversible cell injury. Okay, reversible cell injury. Now you already know it. Now in reversible cell injury, what happens when you take out the stress? Cell will go back to its normal physiological activity. Okay. So in reversible cell injury, what happens? First, let's begin with the hypoxia. The major culprit is hypoxia. Okay, the main problem is with the hypoxia. See, now hypoxia, what it will do? So whenever oxygen is not available, hypoxia means oxygen is not available, right? There is less oxygen availability. Do you know who is going to first get affected? It's a mitochondria. So there will be mitochondrial dysfunction. There will be mitochondrial dysfunction. So mitochondria is getting affected. Now when the mitochondria is getting affected, now tell me what happened to the ATP levels. ATP levels will go down. So there is decrease in the amount of ATP production. Now in your exams they will ask you, what is the first change seen in the cellular injury? Whenever the cells are getting affected, damaged, what is the first change that is seen? So this is the question, sir. First change in cell injury. The first change in the cell injury is mitochondrial dysfunction. Okay. Now what happens when the ATP is going down? Now this ATP is very much important, okay, for most of the things in our body, for most of the reactions, for most of the things in the body, the ATP is very much important. But I want you to know on each and every cell surface, okay, in our body, let me draw here, say this is the cell, okay, I am showing you here, this is a cell. On the surface of the cell, there is a transporter present, okay, there is a transporter which is present over here. So this transporter, by using the ATP, okay, by using the ATP, what it will do is, it will take 3 sodium into the cell, sorry, it will take 2 potassium into the cell in exchange for 3 sodium. Okay, first sodium will go out and potassium will come in. So the name of this transporter is called as sodium potassium ATPase. Sodium potassium ATPase. Okay, sodium potassium ATPase. So each and every cell sodium potassium ATPase is there, and what it is doing continuously 24/7 by using the ATP, it is throwing sodium out of the cell and bringing the potassium into the cell. It requires ATP, ATPase, primary active transport. Now, whenever there is hypoxia, mitochondrial dysfunction, ATP production is getting affected. Now, ATP is not going to be available for this transporter. The ATP is not going to be available for this transporter to transport these substances. So, what happens? These transporters are not going to work. So, what happens? The sodium which was supposed to come out, the sodium was supposed to come out, 3 sodium was supposed to come out, is now not going to come out. So, what happens? The sodium levels are going to increase inside the cell. So, intracellular sodium concentration increases. Which is good? No. Wherever there is sodium, sodium attracts the water. Wherever you have the sodium, so from the extracellular compartment, fluid will shift into the intracellular compartment. Water will try to enter into the intracellular compartment. So now the cells are getting swollen. So what is the first morphological change? See, the first change is mitochondrial dysfunction, which cannot be appreciated under the microscope. You cannot appreciate whether the mitochondria are working or not. Okay? You cannot know whether the ATP is getting produced or not under the microscope. What you can see under the microscope is the histology, histological changes. So, the first morphological change under the microscope is hydropic swelling. Okay, hydropic change or hydropic swelling, the cells are going to swell up. Okay, the cells will take the water and they are getting swollen. So this is called as the hydropic change, which is the first morphological change that is seen with the cell injury. Now the second point which I want you to know is, as a cell is getting this water, okay, as a cell is taking up this water, in order to accommodate this water, okay, in order to accommodate this water, do you know what exactly happens? See, the cell membrane will form the blebs to accommodate the water, otherwise the cell will simply blast. So to prevent that, the cell is going to form this extra pouches like this. So what I am discussing here, the cells are going to get this blebs. So these blebs are called as cytoplasmic blebs, okay, or cellular blebs. Cytoplasmic blebs, which are also seen with the reversible cell injury, okay, which are seen with the reversible cell injury. Next point which I want you to know is, see, this is the first event. The sodium potassium ATPase are not working. Because of that, the water will try to enter into the cell, which is called as a hydropic change, which is the first morphological change. The second thing which I want you to know is, as the water is coming into the cells, even the cell organelles will swell, okay, even the cell organelles are going to swell up. The endoplasmic reticulum is going to swell up, the Golgi complex is going to swell up. See, the endoplasmic reticulum, especially the rough endoplasmic reticulum, is attached with the ribosomes, right? See, for example, if this is the rough endoplasmic reticulum. Now, the rough endoplasmic reticulum is attached with what? Ribosomes, which are involved in the protein synthesis. Now, as this water is coming up, the endoplasmic reticulum is going to swell, which will lead to detachment of the ribosomes. Second event is detachment of ribosomes. Why this is happening? Why the ribosomes are getting detached? Because of swelling of endoplasmic reticulum. Swelling of endoplasmic reticulum. So this will ultimately lead to decrease in protein synthesis. Okay, this will lead to decrease in the protein synthesis. Now, after this, what else you should know is this patient. Okay, so this person, the cell which is undergoing reversible cell injury, the cell membrane is going to get damaged because of the influx of water. The water is coming, right? The cell is getting swollen. Okay, the cell is getting swollen. The cell organelles are also getting swollen. Our cell is surrounded by the cell membrane. So because of this distension in certain part, the cell membrane is going to get damaged. The phospholipid bilayer is going to get little stretched out and it's going to get damaged. Even there is this membrane, okay, even the phospholipid membrane is also present around the cell organelles. Even your cell organelles are membrane bound. There is membrane, this phospholipid membrane is present around the endoplasmic reticulum too. It's present around the Golgi complex. It's present around the mitochondria. So this membrane is going to get damaged and it's going to form a round whorl-like structures within the cytoplasm. So if this is the cell, what you will see is inside the cytoplasm, you will see the structures which are having these whorls, which are nothing but these phospholipid membranes. So these structures are called as myelin figures. Myelin figures. Okay, these are called as the myelin figures. So these myelin figures, what exactly are they? I have explained you. These myelin figures are nothing but cell membrane. Okay, they are phospholipids. Okay, cell membrane is made up of phospholipids. It's the phospholipids within the cell membrane are getting a little damage. The cell membrane is getting damaged. These phospholipids are getting leaked and they are forming this whorl-like structures. Okay, because they are hydrophobic things, right? These lipids are the hydrophobic things, so they are forming this whorl-like structures. They are going to get curled. Okay, so these phospholipids and also they contain some amount of calcium. So this is the question which was asked in the exam. Myelin figures, they are made up of what? The myelin figures are made up of phospholipids as well as calcium. And my question to you, myelin figures are seen in reversible cell injury or irreversible cell injury? Both. Seen in both reversible and irreversible. But usually whenever we say myelin figures are present, usually most of the time we are talking about the reversible cell injury. Of course, they are also present in the irreversible cell injury. Irreversible cell injury means the cell is dying, more damage is going to occur. Of course, you will see the myelin figures. But usually speaking, when I am talking about myelin figures, most of the time I will be talking, I am referring to the reversible cell injury. Okay. So, done. Next, what else you should know in the reversible cell injury? See, as the ATP is not available, okay, as the ATP is not available because oxygen is not there. Okay, ATP production is going down, oxygen is not there. At the end of the day, the cells have to survive. You have to produce the energy. Okay, you have to produce the ATP, but oxygen is not available. So, cell will shift from aerobic glycolysis into anaerobic glycolysis. As there is no availability of the oxygen, now cells are shifting into anaerobic glycolysis. Okay, anaerobic glycolysis. The problem with the anaerobic glycolysis is, yeah, ATP is getting produced. Even in anaerobic glycolysis, ATP will be produced. But lactic acid is getting produced. Okay, lactates are getting produced. So lactic acid, it's an acid. So because of this lactic acid, what happened to the pH? The pH is going to decrease. There is acidosis. Now there is acid environment. The pH is getting decreased. Now because of this decrease in the pH within the cells, okay, within the cells there is clumping of chromatin. So this is very much important for your exams. What is the nuclear change that is seen? See, the nuclear change that is seen in reversible cell injury is the clumping of chromatin. First morphological change that is seen, it is the hydropic change or cellular swelling. What is the first change? It is the mitochondrial dysfunction. There is clumping of chromatin, myelin figures, protein synthesis decreases, okay, cell membrane damage. So all these, cell membrane damage but little, not too much, just normal. So all these are the events which are seen in the reversible cell injury. Now after reversible cell injury, now let's discuss about irreversible cell injury. Now when the cell is getting damaged beyond repair, too much damage, then it is no longer reversible. Now it is irreversible, which means it is imminent, cell have to die. So now let me show you here. First, reversible cell injury. Now it's going to become irreversible. Cell injury. Reversible cell injury and irreversible cell injury. My question to you, when you are going to say that the cell is now damaged irreversibly, okay, now the cell is totally irreversibly damaged. Now what is that thing, what is that morphological thing which will say, now sir, this is irreversible cell injury, the cell will die for sure. See, whenever there is severe cell membrane damage, now whenever there is severe cell membrane damage, or you can say severe mitochondrial damage, severe cell membrane damage or severe mitochondrial damage, once the mitochondria are damaged too much, once the cell membrane is damaged too much, now it is 100% sure that cell will die for sure. It is irreversible cell injury. Okay, and even some books mention severe damage to nucleus. Severe damage to nucleus beyond repair. Now whenever such events happen, severe damage to nucleus, severe damage to cell membrane, severe damage to mitochondria, whenever such events happens, now the reversible cell injury is going towards the irreversible cell injury and the cells will die for sure. Now what exactly happens, sir? Okay, the cell membrane is getting damaged. Now if you look here, this is the cell membrane is getting damaged in multiple places. Let's put it this way, this is the cell. Now if the cell is getting damaged, something like this, what happens? From outside, calcium will come into the cell. Okay, the calcium concentration inside the cell is very less, the calcium concentration outside the cell is more. So from high concentration to low concentration, automatically calcium comes into the cell. Now important point is that this calcium, where it will go? The calcium, it will go into the mitochondria. Even mitochondria is damaged. So the first calcium will likely to go into the mitochondria. Calcium likes the mitochondria. So where this calcium is going? Calcium is going into the mitochondria and calcium is settling within the mitochondria like this. So what you are seeing? So the mitochondria which is having lots and lots of calcium. So these are called as large flocculent mitochondrial densities. Okay, large flocculent mitochondrial densities. Now which are nothing but the calcium deposition within the mitochondria. So these are hallmark. Okay, so what are the hallmark of irreversible cell injury? It is this calcium deposits within the mitochondria. That's a large flocculent amorphous mitochondrial density is their hallmark of irreversible cell injury. And one more thing which I want you to know is, see, calcium is an enzyme activator. Okay, calcium, it is enzyme activator. Normally within the cell, the calcium levels are kept very, very less. Okay, actually the calcium within the cytoplasm is very, very less. Now once the calcium enters into the cell, inside the cell, do you know what this calcium is going to do? The calcium will go and activate the enzymes, they will activate the proteases, they will activate the nucleases. Okay, the proteases, nucleases are going to be activated. So it's an enzyme activator. Okay, it activates the enzymes. Because of that, so the activated enzymes are going to cause the destruction of the proteins within the cell, destruction of the nucleic acid within the cell, destruction of the phospholipids. The membranes are going to get damaged because of the influx of the calcium. Okay, this is the point which I want you to know. Next, once the cell it's decided that it's going to die, it's an irreversible cell injury. Do you know what are the changes that are going to happen in the nucleus? Now in reversible cell injury, because of the change in the pH, there is clumping of chromatin. Okay, there is clumping of chromatin. But in irreversible cell injury, what happens is, first the nucleus, it will condense. Okay, it will condense. It will actually becomes small in size. Okay, condensation, not the clumping, condensation of the nucleus. The nucleus is getting smaller and smaller and smaller. So this process is called as pycnosis. Pycnosis means what? Condensation or shrinking. So followed by pycnosis, now the nucleus is going to become smaller and smaller. It is getting into smaller and smaller pieces. Okay, pycnosis is the nucleus is getting condensed, small, and next the nucleus is going to get broken down into smaller and smaller pieces, into small fragments. This is called as karyorexis. Okay, karyorexis. So karyorexis means what? Karyorexis means fragmentation. Okay, fragmentation. Now after karyorexis, there will be karyolysis. Karyo means nucleus, lysis means what? Dissolution. All the small fragments are going to be totally dissolved. There is going to be dissolution. Okay, dissolution of the nucleus. So pycnosis, karyorexis as well as karyolysis. So these are the nuclear changes. What are the cytoplasmic changes which you can see in the cytoplasm? There are mitochondria with large amorphous flocculent densities, which are nothing but the calcium. Okay, calcium deposition. So why the calcium is coming into the cell? Because of the damage to the cell membranes. Calcium is an enzyme activator. Once it comes into the cell, it activates different enzymes which can cause the damage to the proteins, which can cause the damage to the nucleic acids. Okay, done. So reversible cell injury and irreversible cell injury. Once the irreversible cell injury is there, so first nucleus is going to become pycnotic, pycnosis, karyorexis, karyolysis. Now after this, what happens is cell is going to die. Once first nucleus will go away from the cell, now the cell will die. So the pattern of cell death. After the cell death, how the tissue is looking under the microscope, whether the cell architecture is preserved or not, whether there is total complete liquefaction. So based on the type of pattern, based on the type of histological appearance, the cell death is divided into different types. That is necrosis, apoptosis, pyroptosis, necroptosis, that we will discuss in the next video. Okay, in the next video we will see the different types of necrosis followed by what is apoptosis, necroptosis and pyroptosis. Hope the video is helpful. Thank you.